Okay, everybody, in lab today we did some stuff with some genetics and we did some stuff with some Punnett squares. So I wanted to make sure that everybody was all on the same page with this. So let's say we have this big B, little b individual reproducing with this little b individual. What would you call this individual? What would you call this guy here? Okay. It has two different kinds of alleles. It has one dominant and one recessive allele. So you would call this individual a heterozygous individual. So its genotype is heterozygous. Its phenotype is just going to be dominant, remember. The only thing that you're going to be able to see is that dominant allele expressing itself. These individuals over here have two recessive genes. They have two recessive alleles. So what would you call these? Well, I would call those homozygous recessive. Okay, and again we're sort of crossing them with one another. So you put the gametes that they can create, and the gametes that this one can create over here are just like a B, one big B, and a little b, and the gametes that this one can create are just like a little b and a little b. So you make your Punnett square. And you wind up with big B, little b, big B, little b, little b, little b, and little b, little b. So the offspring from this cross were two heterozygous individuals and two homozygous recessive individuals. Notice there are no heterozygous dominant. And you can actually, this has some predictive power. You can use the alleles of individuals in a population, whether they're heterozygous or homozygous, and figure out how many you expect just by chance there to be in subsequent generations. So it gets a bit more complicated than that. One of your questions from your textbook, or from the lab rather, was asking you to cross an individual with two different alleles that were homozygous dominant and an individual with two alleles which were homozygous recessive. So the way you kind of need to start doing that is to think about this individual here. Okay? Think about which alleles, or I'm sorry, which gametes this individual can actually produce. And the way that you do that is kind of just by foiling it or looking at all the different combinations of alleles that it can have. So we'll start by looking at this one here. So it can have a big F and a big T. And we'll just take that and represent it here. This is what a human egg looks like. So this is one gamete that this organism can create. We'll do another comparison. We'll look at another two letters that it can have. And you can see this is another gamete that it can create from this combination. Let's look at our third combination here, and you'll notice it's exactly the same two letters, the big F, the big T. And finally, the fourth combination here, you wind up with a big F and a big T as well. So those are all the gametes that this individual can create. We'll stick them off to the side while we think about the other organism. What kind of gametes can it create with this homozygous recessive genotype? And again, if you just start looking here, you notice a little f and a little t. So we'll put a sperm here to represent that combination. This is one gamete that it can create. We'll look at another combination, and again, it's a little f and a little t. So we'll put that sperm here. Our third combination is, again, little f and little t, and it should come as no surprise to you that our final combination is little f, little t. So when you have these homozygous individuals, the only alleles that they can create are homozygous alleles. In the case of the female party, this was homozygous dominant. In the case of the male party here, this is homozygous recessive. This is not even remotely to scale, by the way. The sperm are way, way tinier than the egg, but that's fine. So if you have a homozygous individual for two alleles or three alleles, the only kind of gametes they can produce are homozygous. So again, we're trying to cross these two things. That is our ultimate goal. And so what we can do is take those gametes that we've created and put off to the side here, and we can take our friend the Punnett square, and we can line up all of these different gametes with the Punnett square, and I filled out this first one here, because this is a combination of this big F and this little f, and this big T and this little t. And you'll notice that these look the same everywhere. They are all what is referred to as heterozygous. So if you go through and you figure out, okay, well, this is going to be a big F, put that there. This is going to be a little f, we'll put that here. This is going to be a big T, so we'll put it down here. This is going to be a little t, so we'll put it down here. If you combine heterozygous dominant and there is no heterozygous dominant, pardon me. If you combine homozygous dominant and homozygous recessive, the result will always be a heterozygous individual. All right, so homozygous dominant, homozygous recessive, you cross those, you always wind up with heterozygous individuals. So all of the offspring of this cross will be heterozygous. The next thing that it wants you to do is grab a couple of these individuals. So I just sort of grabbed this one, and it wants you to make a cross. So it wants you to make a cross of these two heterozygotes. All right, so two individuals from this generation, it wants you to cross those again. So again, the first thing you need to do is figure out, well, what kind of gametes can this individual make? And so we just start looking at the various comparisons. And I didn't really do this before, but I find a good way to do it is to FOIL it. So here we're doing our first comparison. If you remember from math, this is kind of how it works. And that's going to give you this genotype here for this egg, big F, big T. Then we're going to do this other comparison here. This is just, we're looking at all the combinations that you can get, and that gives you big F, little t. Okay. First, outside, for those of you that remember from math. The next comparison in our FOIL is going to be inside, which is going to result in a little f from this and a big T from this. And then finally, our last comparison to out there is going to give us a little f and a little t, just like that. And the exact same process can be done for sperm. It's the exact same kind of foiling idea where you're just saying, okay, what are all the combinations of these two alleles that I can wind up with? And the sperm and eggs of a heterozygous individual are going to ultimately wind up looking the same. So again, we're going to take all of our eggs from those individuals and all of our sperm from those other individuals, and we're going to cross them, and we're just going to see what all combinations you can get. So you get big F from here, big F from here, big T from here, big T from here, or big F from here, big F from here, big T from here, little t from here, and so on until you filled out the chart. And you'll notice that there are certain patterns, and these patterns, your lab was trying to get at them. They showcase the number of expected individuals in the next generation. So if you're really only expecting 1 out of 16 to be fully homozygous dominant, and I messed, no, it's just one, and I messed this one up over here, but that's okay. You get the point. The point is you wind up with certain ratios of individuals in the next generation that you expect, and that's really what your lab was trying to get at.